Have you ever wondered why some people perform very well under normal circumstances, but crumble under pressure? Or why some people can't seem to get their gears in motion in day-to-day tasks, but somehow manage to switch it on when the chips are down? The reason why has to do with how people respond to psychological stimulation, also known as arousal. According to the inverted U hypothesis, there is an optimal level of arousal for performance, with levels of arousal either above or below this sweet spot, resulting in suboptimal performance. The name of the inverted U hypothesis comes from the shape of the relationship between arousal and performance. Performance increases with arousal up until a certain point, after which performance begins to decline as levels of arousal continue to increase. This relationship was first discovered in 1908 by the psychologists Yerkes and Dodson in experiments involving maze solving in rats, and has since been replicated countless times in humans in tests of athletic performance as well as problem-solving tasks. Arousal is determined by the levels of dopamine in the prefrontal cortex. During tasks which are not particularly stimulating, there is a lack of dopamine in this brain region, which is experienced as boredom and lack of engagement. On the opposite end of the spectrum, a task which provides too much stimulation will overload the prefrontal cortex and result in poorer performance due to anxiety and stress. The sweet spot of stimulation between these two extremes causes feelings of engagement and results in optimal performance, widely referred to as the flow state. But the optimal level of arousal is different for each individual, and this depends on individual differences in baseline levels of prefrontal cortical dopamine. There is a gene called catechol O methyltransferase, or COMT, which codes for an enzyme which regulates the baseline levels of prefrontal cortical dopamine by breaking down dopamine into smaller molecules ready to be transported out of the brain. There are two well-studied variations of this gene which have come to be known as the worrier variant and the worrier variant. The worrier variant is the evolutionarily older version of this gene. It is the most efficient version of the gene and therefore clears more dopamine out of the prefrontal cortex. The worrier variant is an evolutionarily newer variant and, unlike the worrier variant, is unique to humans, being absent in all other primate species. The variant is a change in a single base of DNA, which in turn results in a change in one of the amino acids which makes up the COMT enzyme. The result is an enzyme which is at least 40% less efficient at breaking down dopamine, although some estimates put this as high as 3 to 4 times less efficient. As with all genes, a person inherits one copy from their mother and another from their father, so there are 3 possible combinations of this gene in any given person. Homozygous warriors, that is, both versions of the COMT gene are the worrier variant. Homozygous warriors, that is, both copies of the COMT gene are the worrier variant. And heterozygotes, that is where you have one copy of each version of the gene. Baseline levels of dopamine in the prefrontal cortex depends on which combination of variants you have, with more worrier variants resulting in higher baseline levels of prefrontal cortical dopamine. Having increased baseline levels of prefrontal cortical dopamine means that it takes less stimulation to reach the optimal level of arousal for performance. People with higher baseline levels of prefrontal cortical dopamine have an advantage in that they have better working memory and executive function in low arousal activities such as tackling unsolved mathematical theorems or filling out their taxes correctly. The disadvantage is that high arousal situations are more likely to push warriors out of their comfort zone, and they experience the drop in performance visualised by the right hand side of the inverted U. Persons with lower prefrontal cortical dopamine levels at baseline show lower performance in the low arousal activities in which warriors are at their peak, as they struggle to maintain their attention in tasks which do not provide them with sufficient arousal. Boredom comes quickly and distraction beckons with her siren's call of junk food and video games. However, when the going gets tough, the warriors get going. High arousal activities which would paralyse the warriors, such as tackling armed bank robbers or sinking a buzzer-beating basket at their community basketball league, are where warriors excel. Prefrontal cortical dopamine is associated with the trade-off between cognitive flexibility and cognitive stability. Cognitive stability refers to the ability to concentrate on the task at hand without distraction from external stimuli or competing thoughts. In contrast, cognitive flexibility refers to the ability to quickly switch between tasks in reaction to external events. Carriers of the worrier variant have been shown to be more successful in tasks requiring of sustained attention, whereas carriers of the worrier variant have been shown to be more successful in tasks requiring switching of attention. One example of this is the difference in performance between worriers and worriers in an experiment called the Wisconsin card sorting task. Participants are given a set of cards containing pictures of shapes. Participants have to sort the cards according to a rule, which could be that the cards are to be sorted by shape, sorted by colour, or sorted by the number of shapes. They are not told the rule in advance, however, after sorting the cards they are given feedback about whether they have sorted them correctly and given a new set of cards to sort. Participants are given a new set of cards to sort each turn and continue to sort cards once the correct rule is established. This continues until, on a random turn, the rule is switched and participants must re-establish the new rule. Worriers proved to be more efficient at sorting the cards once the new rule is established, a task demanding of cognitive stability, but worriers were faster to establish new rules, a task requiring more cognitive flexibility. Accordingly, carriers of either variant thrive in different walks of life. Worriers have been shown to perform better on the SAT, a test of academic ability required for admission to US colleges, and are therefore presumably overrepresented in careers requiring a university degree. Worriers, on the other hand, are overrepresented in entrepreneurial careers. There is also a higher frequency of homozygous worriers amongst professional athletes, especially mixed martial arts fighters, suggesting an advantage conferred by their increased performance under stressful conditions. So why are there two versions of this gene in the first place? The fact that this variation in the COMT gene has not been found in other primates, our closest evolutionary relatives, suggests a possible role in the evolution of human brain function. One hypothesis is that in human populations which transitioned from a forager's lifestyle to agricultural societies, the adoption of more specialised roles has induced selection for genetic propensities which are better adapted to tasks which are repetitive and demanding of sustained attention, compared to the lifestyle of a hunter-gatherer for which the ability to switch attention more rapidly is better suited. This hypothesis is supported by the fact that the frequency of the worrier variant is lowest in populations which subsist at least partially by hunting and gathering. The highest frequency of worrier variants is found in populations of European ancestry, where there is a roughly equal frequency of both variants, which is indicative of balancing selection, meaning that worrier-worrier heterozygotes have been more reproductively successful in this environment than homozygotes of either type. The naming of the worrier and worrier variants come from the pathologies associated with each genotype. Carriers of the worrier variant are more likely to display aggressive behaviours, use illicit drugs, and are at an increased risk of schizophrenia. Interestingly, there is also some evidence to suggest that they are more susceptible to hypnosis. Homozygous worriers, on the other hand, are at an increased risk of anxiety, depression, and PTSD, have lower emotional stability, and a lower pain threshold. Women with anorexia or bulimia are more likely to carry the worrier variant, and there is some evidence to suggest that carriers of the worrier variant are at an increased risk of obsessive-compulsive disorder. But worrier and worrier variants don't deserve this bad press. No one is doomed to become a violent criminal or a nervous wreck because of their genes. Instead, it is more accurate to think about COMT variants as modulating how a person develops in response to their environment. For example, it has been shown that homozygous worriers are more likely to display aggressive behaviours only if they have experienced violence themselves at a young age. Similarly, carriers of the worrier variant are only at an increased risk of anxiety disorders if they have experienced a traumatic event. This characterisation of the negative aspects of the COMT variants reflects a broader overall tendency in psychology to focus on the pathologies rather than the strengths of individual differences in personality. And this only tells us half the story. Worriers thrive in situations which require responding quickly to aversive stimuli and keeping cool under pressure. Worriers, on the other hand, are better at solving cognitively difficult problems. Considering these aspects of the COMT variants, worriers and worriers could just as easily be rebranded as heroes and philosophers. Much more positive and romantic, although admittedly lacking the alliterative quality that worrier has. So, will the version of the COMT gene which you carry determine your personality, career and life choices? The answer to that is no, at least not completely. The effect of COMT on all of these things is real, and these associations have been replicated multiple times by different researchers, but that doesn't necessarily tell you much about any specific individual. If you look at the distribution of scores for this test of cognitive stability and cognitive flexibility, for example, you will see that although there is a significant difference between the means, there is a big overlap within the confidence intervals between carriers of all three COMT genotypes. This is because the worrier variant is only one of the many factors which influence levels of dopamine in the prefrontal cortex. Although the worrier variant affects the efficiency of the COMT enzyme, this is not the only genetic feature which controls COMT activity. Just like any other gene, there are genetic elements called promoters and enhancers which control when the COMT gene is expressed and how many copies of the enzyme it produces. And these elements can come in different versions with differing levels of activity. So, for example, even if a person carries the worrier variant, they may have promoters for this gene which are underactive, giving them essentially worrier levels of prefrontal cortical dopamine. Also, other biological factors not controlled by COMT influence the amount of dopamine produced in the first place before COMT degradation. There are also developmental factors which influence the development of the prefrontal cortex. For example, environmental deprivation or poverty can impair the functional development of the prefrontal cortex, and this will moderate the contribution of COMT variation to individual differences in behaviour. While a range of genetic and environmental factors influence the development of how the prefrontal cortex responds to arousal, cultural factors influence what qualifies as an arousing event in the first place. Snakes may cause people to jump on tables and scream in some places, whereas in other parts of the world they may simply be a household nuisance. Researchers are only able to detect a reliable difference in behaviour between carriers of different COMT variants when using large sample sizes. By grouping participants based on their COMT variant status, you increase the likelihood that the other factors which influence prefrontal cortical dopamine balance each other out. So, although sequencing a person's COMT gene may not tell you much about that person, COMT is still a useful candidate gene for researchers to influence the effect of variation in prefrontal cortical dopamine on behaviour. Learning about your own COMT variants can still be useful for understanding yourself. If you have trouble concentrating on tasks which you find to be boring, it doesn't mean that you are lazy or stupid. You may simply have lower levels of baseline prefrontal cortical dopamine and will excel in tasks which you find more stimulating. Similarly, preferring crossword puzzles over skydiving doesn't make you a buzzkill. Your higher baseline levels of prefrontal cortical dopamine make you a master problem solver. Whether you are a worrier, or a carrier of both variants, you will flourish once you find the right environment for your genes, brain, and personality.